Hi everyone, thank you for tuning in. We're excited to tell you about our course innovation grant entitled Digital Storytelling in Biology, Increased Engagement and Inclusiveness Through a Novel Assessment Method. And our grant was submitted by myself, Ariana Smith, and Wade Powell. Our course innovation was implemented in a course called Bio 116, Information and Living Systems, which is the second part of introductory biology at Kenyon. And every spring, we host somewhere between 70 to 100 students across two sections of the course. And the enrollment in the course reflects the increasing racial, ethnic, and socioeconomic diversity in the Kenyan student body more generally. And it also hosts mostly first years and sophomores who have varying interests and uncertainty about their pursuit of a science major. And so the increasing diversity means that we have students with different levels of preparation coming into the course. So we as instructors of the course certainly feel that it offers the most exciting content in molecular biology, genetics, and evolution, but we realized that there were structural features of the course that could pose as barriers to inclusion. And evidence for that statement is depicted in this graph that shows the distribution of scores on the first exam of the semester in one section in spring 2017. And in this section, there were 9 students who would identify as students of color. And of those 9, more than 75% of them earned a grade of C- or below. So to address these potential barriers to inclusion in our classrooms, we went to the experts and we attended the Best Teachers Institute, which is founded by Dr. Ken Bain, who is also the author of a book called What the Best College Teachers Do. And in this book, Ken Bain notes that students take a deep approach to learning when they are solving problems that they find intriguing, interesting, or beautiful. So we turned our attention to the assessment activities that we feature in Bio 116, which include 10 weekly quizzes, 4 exams, and a comprehensive final, and asked very simple questions like, is it intriguing or interesting or beautiful? And the verdict was no. None of these assessment activities meet any of these criteria. And so we concluded that despite a wealth of course material that is featuring the most exciting challenges in molecular biology and genetics, we weren't doing enough to engage students in these very intriguing, interesting, and beautiful ways. So on the heels of the Best Teachers Institute, we sought to develop a course capstone project that allowed students to ask and answer questions in biology that were intriguing, interesting, and beautiful, and allowed them to display their conceptual knowledge with us in ways that were unique and non-traditional. And to this end, we developed the Digital Storytelling Project, which was also supported by the Center for Innovative Pedagogy. And this project focused on CRISPR-Cas9, which is an exciting new technology because of its applicability in editing the human genome. But in order for students to understand how CRISPR-Cas9 works, you have to have a fundamental understanding of concepts of basic concepts in genetics and molecular biology. And this is the exact content of Bio 116. So in this project, we asked students to identify a heritable disease and develop a CRISPR-Cas9 based strategy that could be used to treat or cure this disease. Implementation of the project in the course required significant restructuring and in some cases condensing of course content in order to accommodate 3 new in-class workshops. And the intention of these workshops was to familiarize students with really sophisticated bioinformatic and literature databases. And the students would use these databases to explore genetics and molecular features of a chosen heritable disease. And the assignment was scaffolded across the semester. So after each workshop, the students would generate what we call storyboards, and we evaluated those storyboards and offered feedback before the storyboards were condensed into a short video. In workshop 1, students were asked to identify and describe a heritable disease. And in this example storyboard, you can see evidence of that here for a disease called retinitis pigmentosa, which is a progressive vision loss. You can see that the students are outlining the phenotypes in the eye and also outlining what the world looks like to someone who has retinitis pigmentosa. In addition to characterizing the disease, they were also asked to determine the mode of inheritance and determine the genes involved. And so you can see how they incorporated this into their storyboard by pointing out the location of the gene on the chromosome and showing us the protein structure for the protein encoded by that gene. In the second storyboard, students use another sophisticated set of databases to provide information about the gene structure, which they show here, the protein structure and function, which they indicate here, and then also mutations that are present in patients with the disease of interest, as they highlight here. Finally, the students share a strategy for repairing the mutated DNA sequence with a corrected DNA sequence, and that's really clearly outlined right here in this storyboard. In the third and final workshop and storyboard, students were asked to scan the primary literature to look for evidence of when and where the gene was expressed to inform when and in which cell type or tissue type they needed to deliver their gene therapy. At the end of the semester, students were asked to critically evaluate each of their storyboards, incorporating feedback from their instructor to select the most relevant materials for describing the genetic disorder, characterizing the genetic and molecular underpinnings of this disorder, proposing a CRISPR-Cas9 based strategy to treat or cure it, and then finally to provide a compelling argument for or against the designed therapy. And students took this collection of materials and organized it into a digital story that was assembled using VoiceOver PowerPoint, and each video spanned somewhere between 3 to 5 minutes. The final digital story then took the place of a cumulative final exam. From an instructor perspective, we felt like students made deep investments in the project and that the incorporation of the project, and particularly the bioinformatic and literature databases, really expanded upon and even challenged the students' conceptual understanding of content in genetics and molecular biology. At the end of the semester, in course evaluations, we asked students in both sections of the course in the spring of 2019 to rate the effectiveness of the digital storytelling project in reinforcing basic content learned in the class, building their scientific skill set, developing their abilities to function as a citizen scientist, leveraging their creativity in scientific context, and also enjoying the learning process. And generally, the students ranked the digital storytelling project as being quite effective in each of these areas. And this was impetus for us to try it again. Successful completion of the digital story requires that members of a group be in routine communication with one another across the semester. And we recognize on the onset of the project that there could be considerable challenges with group work. Thankfully, in the spring of 2019, when we implemented the project for the first time, we had an uneven distribution of students in Section 1 versus Section 2, such that we were able to take advantage of a natural experiment. So in Section 1, we formed groups that had 2 students in each group, and in Section 2, we formed groups that had 4 students in each group. And at the end of the semester, in course evaluations, we asked students to tell us open-endedly how we might improve the project. And then we coded those answers from both Section 1 and Section 2. And so what you see here is, of the responses, a large fraction of students in Section 1 and Section 2 recommended that we alter the group structure. And so in subsequent semesters, we transitioned each group to having 3 students per group. And anecdotally, we learned that transitioning the group size to 3 was actually a favorable and helpful modification, and something will likely continue in future iterations of the project. We believe that the digital storytelling project outlined here is a worthy addition to the Bio 116 course structure. It's one that allows students to be creative in how they think about and apply the course content, and allows us to assess their learning in ways that are not conventional. Some considerations for the future surround the nature of the course itself. For example, Bio 116 is a course that rotates around professors in our biology department. And so we would like to be considering expanding the project beyond instructors, Professor Wade Powell and myself, who developed it, and even beyond human disease and beyond CRISPR-Cas9, to be more compatible with the skill set that different instructors in our department might have. So this project was funded by the HHMI Inclusive Excellence Grant, the Digital Storytelling Project Fund from the CIP. And we would also like to acknowledge Professor Toshi Tsunekage for his contributions to the project in the spring 2020 semester.